the first place. Okay, so remember that the whole concept of exchange revolves around the idea, half an hour starts now, of respiration. Respiration. So all the cells in the body of an organism need to carry out respiration. And for that process, so what's the point of respiration? Why do we do respiration? It makes such a big difference having an audience. Look at that, all that interaction going on.
Produce ATP.
Thank you. Right, ATP. So because the cell needs to carry out lots of activities, anabolic reactions, active transport, things like that, so the cell can't live without some of its metabolic reactions, and a lot of those metabolic reactions need ATP. So we need ATP, and ATP comes from respiration. But respiration itself needs an input of glucose, right? An input of oxygen gas, right? And I'm just going to add in there nutrients, just general nutrients, because it's not all about respiration. You need materials to build the cells, etc. And as a consequence of this, right, the glucose gets broken down to CO2. Right, we produce water, but that's not really a problem. But at the same time, we produce metabolic wastes, such as urea. Okay, so all organisms need to carry out exchange because they need certain things from the environment, being oxygen and being nutrients and being glucose for respiration, but they're also producing wastes. So we can't forget about that. They're producing waste that they're going to have to get rid of. Yeah? Okay. I think the next part of this is the mechanism. So how exchange happens. How long do you think it's going to be before I fall over this one?
Yeah,
I've already
I'm actually worried.
intertwined myself quite a number of times. Okay, so how exchange happens. So the whole point here is exchange happens by way to show what a great teacher I am, guys. Exchange happens by, all right, fine. You know what? Fine, I'll just pretend like you're not here. The camera's not here, you're not here, I'm by myself. I'm teaching nobody. Right. How exchange happens is by diffusion. So whether you're a single-celled organism and you don't need a specialized exchange system, or you are a very complex, large organism, either way, exchange happens by diffusion. You get the stuff that you need by diffusion, and you get rid of the stuff that you don't want by diffusion. Okay. Now this happens because of concentration gradients. Concentration gradients. So stuff gets exchanged. It's happening via diffusion. Diffusion is happening because of concentration gradient. Why do we have a concentration gradient? Because remember, if respiration is happening in this cell, then it has a low oxygen concentration inside the cell because it's always using the oxygen up. Okay? I know some of this might be obvious to you guys, but this will underpin the stuff that's going to happen later. So generally speaking, the oxygen availability from the environment, or the oxygen concentration in the environment, is usually higher. So we have a concentration gradient, and that's great. Okay? But as a result of respiration, we're constantly making CO2. So usually, the CO2 concentration inside the cell is going to be higher than the environment, and therefore, again, we have a concentration gradient and CO2 comes out. And it's the same for wastes, and it's the same for other nutrients that might be getting used up by the cell as well. We good? Okay, what's next? Right, the next thing is why exchange surfaces. Why do we need exchange surfaces then? So remember that this all comes down to this thing called Fick's law. Fick's law. Is this ringing bells?
Yeah.
Okay? Now, Fick's law, if we're saying that exchange happens by diffusion, then we need to look at what affects the rate of diffusion, because that's going to affect how quick, how efficiently we're getting stuff and getting rid of stuff. So Fick's law says that the rate of diffusion is proportional. Can you guys see this? Is it too small?
Tiny bit
Tiny
too
bit too
small.
small?
How much are the boxes?
All the more reason for you to watch the video then. All right. But can you not move closer then? Or
Nice.
something?
I just copy. I can't see it.
What about Rabia? Can you see? Rabia can see it. She's behind you.
That's because she has my glasses. She has 20/20 vision.
Zainab? Noria? Okay. Right. So the rate of diffusion is actually, you can just, if you don't see it here, you can just follow along with what I'm saying. The rate of diffusion is proportional to the surface area multiplied by the concentration gradient divided by the diffusion distance. Okay, so yeah, now, so the bigger the surface area, right, and the bigger the concentration gradient makes the rate of diffusion bigger. Yeah? Any questions, do ask me. If you want, I can just cut it out of the video. Yeah? Just make that request. Not that anyone knows who you are. Right. And then finally, the smaller the diffusion distance. So those three things contribute to a high rate of diffusion. If the surface area is big, the concentration difference is big, and the diffusion distance is small. Okay. Right. And what we're saying is, as organisms, so if we have an organism represented by a sphere, as organisms get larger, what happens to their overall surface area to volume ratio? It decreases, it gets smaller, which is what we don't want. The surface area gets smaller. What happens to the diffusion distance? The distance something would have to move to get to all the cells. The diffusion distance gets bigger, right? And because concentrations are building up outside cells, and some cells don't have access to the environment, the concentration gradients are going to get smaller too. So as organisms get bigger, the rate of diffusion, or the effective rate of diffusion, decreases to a point where it is no longer sufficient for all the cells to be able to carry out respiration. And if they can't carry out respiration, they don't have enough ATP for all the metabolic reactions happening in the cell, and so cells die, they don't work properly, wastes build up, etc., etc. Yeah? Yeah, and so, last thing, so far so good. Last thing is, what are the features of exchange surfaces? What are the features of exchange surfaces, therefore? You, Miss Anonymous Person.
What everybody is talking about.
We've said why the rate of diffusion can go down. So what are the features of exchange surfaces that make the rate of diffusion high?
Small surface area. To the volume ratio.
Cut. Okay, you want to go again? Anybody?
Short diffusion.
Right, we want this short as possible. Right, so the key features are, 1, the shortest diffusion distance possible. 2, the highest possible concentration gradient or difference. And 3, the largest possible surface area to volume ratio. Yeah? Okay, and that is important. That is probably the most important thing in the topic. And now what we do is we just apply that to insects, we apply that to fish, and we apply that to mammals to see how they apply this concept of these things. Does that make sense? Okay. I think I did deserve this. By the way, I'm sure if you guys want to eat or drink stuff, I can look away. Gas exchange in insects. Right, off we go then. So the next thing we're going to do is insects. Okay, right, gas exchange in insects. How much space do I have? I'm going to give about that much space to the insects. Okay, so insects. Now, insects, I think we still need to appreciate, because you could easily get a question like this that says, well, why do insects need a specialized gas exchange system? Aren't they really tiny? And though relative to us, they're really tiny, relatively speaking, they are a multicellular, they've got lots of cells. So they are multicellular organisms. Okay? Relative to single-celled organisms, they are large, relatively. Okay? And they're quite active. So I'm going to say metabolically, they're quite metabolically active. So we need to remember this. Okay? Even though that's not obvious to us. Right? And therefore, they need a high rate of diffusion. And so they can't just rely on being small for this. So because they're large and multicellular, effectively what that means is that diffusion distances are going to be large, and it means that the surface area to volume ratio is going to be small. Okay? And that is going to collectively amount to a low rate of diffusion. Okay, so how do insects do it then? So I'm going to draw a very weird insect just to illustrate my point. Okay, so we have an insect there, it's got its 3 body segments. I'm going to ignore the wings and the legs. Okay, and let's just say we've got some cells right at the center of the insect. So how do these cells at the center of the body of the insect, how are they able to get the oxygen that they need and the carbon dioxide? How do they get rid of it? Okay, and what's the structure that allows these cells to continue living or functioning? Begins with S? Yes. Spiracles. Okay. So, remember, so these tubes, they go to the surface of the insect. Okay, so these tubes, do they begin? Are they called tracheae? And then they branch out into tracheoles, right? And then they branch out into, or the spiracles are the openings, and then those openings into these kind of tunnel-like structures called tracheae, and then finally into very small structures called tracheoles. Okay? Now the tracheoles, because they are opening into the atmospheric air, the tracheoles can therefore have effectively a much higher concentration of oxygen, right? So the oxygen can go in there. But why is it going in? Okay, now what are these cells doing? Yep, cells are carrying out respiration, so any oxygen in those tubes, those tracheoles, oxygen is diffusing into the cell. Okay, so what does that do to the oxygen concentration in the tracheole? Goes down. So the oxygen concentration in the tracheole goes down, and therefore, relative to the atmosphere, so now the atmosphere has a higher oxygen concentration than the than the tracheole. And so the oxygen kind of diffuses in. Is that making sense? Okay, so it's not really as active as the lungs, where we're, pushing the carbon dioxide rich air out and we're inhaling the oxygen rich air in. It's not like that. It's more like relying on the process of diffusion, but aiding the process of diffusion by having this network of tracheae and tracheoles. Okay? All right. Fine. Okay. Now, we do need to talk about the three features though, because we shouldn't be talking about this in isolation. We have to connect it to what we discussed before. So, diffusion distance. How is the diffusion distance minimized? Well, you've got these tracheoles, they're quite close to the cells that are respiring. Does that make sense? So the overall kind of diffusion distance is minimized as much as possible because of that. What else we got? Concentration gradient. Well, the concentration gradient is maintained by the constant respiration of the cells, right? So because the cells are respiring, the oxygen is diffusing into the cells. And because the oxygen in the tracheoles is constantly being consumed, the oxygen from the atmosphere is constantly diffusing in. Okay? And surface area. Now, the surface area, because these, there's a lot of branching, highly branched. So because you've got this high level of branching, basically collectively then the tracheoles have a very large surface area, and you're not going to have many cells that are going to be far away from a tracheole. We good? Any questions? Okay, so remember, with the insects, yes, learn the stuff that you're going to learn about, but see if you can connect it up to the central idea, the central concept of the features of an exchange system. Cool? Okay, let's move on. Oh, I did forget something. So some insects can, if they got a greater metabolism, if they got more activity, if they're larger, they can raise and lower their abdomen, okay, which kind of helps the ventilation process, kind of pushes the air in and out a little bit. All right, so what does that help? Diffusion distance, surface area, concentration gradient. Helps to push the air out and kind of suck it in because of the change in volume. What does that help? Surface area, concentration gradient, or diffusion distance? Concentration gradient, right? Because there's going to be CO2 in those tracheoles, and when we reduce the volume of the abdomen, it's going to kind of push that air out, okay? And when we expand that abdomen again, we're going to suck the fresh air into the tracheoles, all right? So we're helping that concentration gradient. Okay, good. Insects done. Let's move on to fish. All right, fish. Fishy fish. Okay, so, right, with fish, right, first, again, with fish, the same concept is going to apply, but we're going to look first at how fishes carry out ventilation. Okay, so we're going to look at that process first. So, are we good? We good so far? All right, so fish ventilation, it's got kind of two bits to it, okay? The first thing is what's happening with the mouth, the buccal cavity, right? Because what the fish is doing is, it's sucking in water, running it past its gills, which is the exchange surface, and it's kind of forcing the water to come in, move over the gills, and then out. Okay? So how does it get the water to do that? So first, the top part of the fish mouth I'm going to keep pretty much the same. Okay? But what it does is it opens the mouth and lowers the floor of the mouth. So it's basically increasing the volume. Okay, so that's step one. Draw it too big actually. Okay, so as it lowers the volume here and opens the mouth, water is flowing in. This is not diffusion. Okay, it's like breathing but with water, right? So it's kind of forcing the water in. All right. At the same time, we also have to be aware of what's happening at the gill valves, okay? What the operculum is doing, gill covering, okay? So at this point, the gill valves are closed, all right? Because we don't want the water, we're increasing the volume here, right? We're increasing the volume here and we're reducing the pressure. We want the water to come in from the mouth. Do we want the water to come in from the gills? No, okay? Because the gills are adapted for one unidirectional flow. It's not both. So while the mouth is open and the water is flowing in here, the water is not flowing in from the gills because these valves are closed, okay? So they're closed. In the next stage, what happens, so the top part I would like to keep the same, but in the next stage what happens, the floor of the buccal cavity is raised and the mouth is closed, so we are reducing the volume in the buccal cavity. What's that going to do to the pressure on the water in the mouth? Increase it. So it's going to increase the pressure here. Okay, but now the mouth is closed, the water can't escape through the mouth, even though there's a high pressure on it. So where is it going to go? Right, it's going to go out through the gills because at this point we have opened the gill valves, okay, and we've kind of moved the operculum so that now the water is allowed to flow out through the gills. Okay, so we've increased the pressure on the water, but given it only one route out of the fish, which is through and over the gills. Okay? And so we get this movement of water, and this effectively is ventilation in the fish. So what of the three features of exchange system is this referring to? Surface area, concentration gradient, or diffusion distance? Concentration gradient. By the constant flow of water, or fresh water, over the gills, we're keeping that oxygen concentration that's going over the gills high. And we're keeping that carbon dioxide concentration low. Making sense? Okay? And again, we're because of that countercurrent that we're going to discuss later, because of that, the gills are adapted, or the gill lamellae are shaped in a certain way, and it needs to be in a certain direction, which we'll discuss. We good? Okay, so steps. You need to know the steps of ventilation, which I'm not going to write them out right now, but the steps of ventilation in bony fish. Okay, so remember to talk about the mouth, what's happening there, and remember to talk about the operculum and the gill valves, or the opercular valves. Right, next. This is going to be now in the fish, the counter current flow. Okay, counter current flow. So exchange at the gills, essentially. Okay, so how? So the gills are like these V-shaped structures, right? So they're like this, right? Like V-shaped structures, right? And the water flows over them, and there's like a stack of them. So I'll draw one, I'll draw another one underneath. Okay. But there's many more than what you can see here. Okay? And the water, because are we agreed that we've got the water flowing in one direction only, it's not two. And that particular direction of water flow, that direction of water flow is this. Over and through the gills. Okay, and this is why it was important to maintain. This is why it was important to maintain that direction. Okay, why it couldn't be in that, out that way, in that way, out that way, in that way. We couldn't have two directions because the whole gas exchange process is most effective when this flow is in one particular direction. Do you guys remember why? Okay, so I shouldn't have done it in blue actually. Right, so just like with the alveoli, we have those nearby blood capillaries. In the same way, the gills is the point in the fish at which exchange between the blood in the fish and the water happens. Right? In the lungs, it's between the air and the blood, but in the fish, it's between water and the blood. So what happens is that a capillary will come in, and it will come in at this point, and it will split and flow through this gill in this direction. Okay? And as it flows, gas exchange is happening. So oxygen is diffusing into the capillary. So as this blood flows, it becomes oxygenated, and then it will exit at the opposite point. Okay, so that capillary will run like this, and as it does so, it becomes more and more oxygenated, and when the blood leaves that gill lamellae, it is oxygenated. Okay? So that's going to happen in that direction as well. Oops. But as it goes, it's going to get oxygenated. Now, what's the big deal about this directional flow? Well, as you can see, the water with the high concentration of oxygen is flowing, hopefully, you can see that it's flowing in the opposite direction to the blood. Are we agreed on that? Yeah? So the blood generally is flowing from the back of the gill lamellae to the front of the gill lamellae, or the blade. Okay, so from the back to the front. Okay. Now, because of this opposing flow, that's what we call counter-current flow, and it ensures that the concentrations, the concentrations between the blood and the water, they never equalize. Yeah, there's always like a concentration difference between the water and the blood over the distance of the gill lamellae. Okay. But the important thing is, that takes a little bit of getting your head around, but the important thing to remember is that the flow of the water is in the opposite direction to the flow of the blood. And that is what helps there to be a concentration gradient over the whole distance that the blood is flowing. Okay then. Structures of the lungs. All right. Okay, so lungs. So if we begin with our airways, right? You do need to know. I might not go. I'm trying to speed things up a little bit. I'm not going to go into every single structure here, but we do need to be aware of, especially. This does overlap with a little bit with module 2, but the lungs is a good example of how tissues are specialized and they work collectively to aid a particular purpose. So in this case, the sole purpose, as with all the exchange surfaces, is the highest possible rate of diffusion. How do we get that to happen? So remember that the airways are kept open because they need to ventilate, right? They need to allow the ventilation to happen. Air going in, air going out, as far as possible, let's not obstruct that process, and let's keep it going as fast as it can, or as fast as requirements need it. So, you've got the airways, and then you've got the lungs. Okay, now, with the airways, what's helping them to keep open? Okay, so the airways, remember, or incomplete rings of cartilage. They, allow those airways to not collapse under the pressure, right? Strength, tensile strength, support, flexibility, okay? As well as that, surrounding these airways, we do have elastic fibers, okay? Elastic fibers to allow a little bit of stretch and recoil as required, and also smooth muscle going around. I'm just showing it in different parts, but you've also got smooth muscle around those airways in the event that you need to constrict those airways. If there's something in the air that we want to protect the body from, we can constrict those airways when required. Okay? Right. Okay, next. Also here, in the airways you've got columnar epithelium. Okay? So again, they're kind of, regular shaped, and they have tight junctions between them so that, the epithelium isn't kind of freely permeable or penetrable to like pathogens and things like that. So you've got tight junctions between the cells, because it's essentially a barrier to the inside of the body. You've also got goblet cells here, right? Goblet cells producing mucus to trap the pathogens to prevent lung infections. Okay, again, the air from the atmosphere that potentially has the pathogens in it, that's coming into the lungs, right? So you need to protect the rest of the body tissues as well as the lungs from those pathogens. And what else? Goblet cells, and go on. Ciliated epithelium. Okay, kind of works with the goblet cells so that anything trapped in the mucus can then be wafted away and keeping those airways clear. Okay, so we need to be kind of aware of the overall structure of the lungs. Right, next we get to the business. Okay, so the airways, the bronchi branch out into the bronchioles, and the bronchioles eventually end in the alveolus. Okay, and again, the theme of branching comes up. The theme of branching comes up, which is related to which key feature of exchange systems? Surface area, right? High surface area to volume ratio. Okay. Yeah, so let's get on with this. Got the alveolus. Again, the alveolus has some adaptations of its own, right? It's got these flat, while the airways has got columnar epithelium. When you get to the alveolus, this epithelium is called squamous epithelium, right? Squamous epithelium. You can see that they're flat. How does that help the function of the alveolus? Right, it reduces or has a shorter diffusion distance, which should effectively increase the rate of diffusion. Then we've got a capillary nearby. Okay, and got the blood cells moving through. Yeah? Okay, so again here we've got a high concentration of oxygen. We've got a low concentration of CO2. And in the blood, it should be the opposite situation. We should have a low oxygen concentration, right? All the deoxygenated blood, oxygen used up by all the tissues, and then arriving at the lungs. But we should have a high concentration of CO2, right? And so we've got a gradient. So the CO2 diffuses in there, and the oxygen diffuses. So the CO2 diffuses into the alveolus, into the air, into the air of the alveolus, whereas the oxygen diffuses into the blood, hemoglobin, etc. Yeah? We good? At this point, we should revisit our key features and just see whether there's anything here that matches up or parallels with the concepts that we've discussed. The squamous epithelium is reducing the diffusion distance. Good. The ventilation, which I guess we'll discuss in a bit, the ventilation, it will be maintaining the concentration gradient just like it does in the fish. The branching and the sheer collective numbers of alveoli is related to the surface area to volume ratio. Okay, anything else? Yeah. So remember, the capillary also has a wall, and careful not to say cell wall, but the capillary has a wall which is one cell thick. So not only are the cells flat, but there's only one layer of cells in that wall, both for the alveolus and the capillary. Okay? And remember that the capillary network, it kind of would be pointless if we had lots of alveoli but not capillaries to exchange with, right? So we want high numbers of alveoli, but each alveolus should have a supply or have a capillary network associated with it. So it's the alveolus and the capillary network which collectively provides a high surface area of exchange. Yeah. Okay. Ventilation? Right, let's talk ventilation then. Hopefully I run out of space before we get to spirometer. Okay, so with the ventilation, it's basically what I want you to just focus on is what's happening during inhale and what's happening during exhale, and the reasons. Okay, the reasons are important. Hopefully what we've done here is focus on the reason for everything, not just the things. Okay, so inhale. Right. The point of the inhale is we want to increase the thorax volume. If we increase that thorax volume, we reduce the pressure until it's lower than the atmospheric pressure, right? And then, because the pressure in the thorax or the lungs will be lower than the pressure in the air will move from the high pressure to the low pressure. It's gradients, it's always gradients, okay? So, increase the thorax volume, that means reduce the pressure, and therefore air moves down the pressure gradient. Air moves down that pressure gradient into lungs. Okay, how did we get that to happen? We had intercostal muscles. I can't remember this. Do they contract or relax at this point?
Contract.
The outer intercostal muscles contract. Outer intercostal muscles contract. Okay, so that kind of raises the rib cage. What else? Diaphragm. Contracts? You sure? All right, all right. Okay. Yeah, all right. So the work of the intercostal muscles and the diaphragm collectively, what that does is it increases the thorax volume. And because the thorax volume increases, it reduces the pressure, okay, in the lungs, and therefore the air goes into the lungs. Okay, exhale, opposite. So we want to force the air out, we want to decrease. We want to decrease the thorax volume and increase the thorax pressure. Okay, and again, air will be, because of that, the pressure in the lungs will then be higher than the atmosphere, and so we're forcing the air out. So the air is forced out of lungs. How did we get that to happen? Well, the intercostal muscles relax. Correct me if I'm wrong. Okay, and the diaphragm also relaxes. Mm-hmm. We good? Okay. Again, the whole point of that is to keep, the concentration. We're maintaining the concentration of oxygen in the lungs high, right? Or in the air in the alveolus high. We're keeping the concentration of CO2 in the air in the alveolus low. Awake? Yeah? Okay. I think that was it, isn't it, in relation to that?
So I always mix up, like inhale and exhale. Like I forgot, like if it's the internal, like external muscles that contract, relax. How do you know?
Yeah. So yeah, that's what is in my head that causes confusion for me also. But I think the outer intercostal muscles contract, and it will be the inner ones that will be relaxing, and the opposite. That's what I remember or think. There's inner intercostal muscles and outer intercostal muscles, and when one is contracting, the other is relaxing, and when one is vice versa. My microphone can pick you up, by the way, even though my ears can't. So. No, just kidding. All right, last thing then, spirometry. It's taking a bit longer than I thought. Do I need to go over the device? Okay, all right. So I'll just go over the device briefly, very briefly, just for the purposes of the video. Okay, now basically there's a tank, okay, and the tank has got water in it. There's a lid on the tank, okay, and basically when people are breathing, there's a little pipe, so there's water, okay, the lid rests on the water, and then there's a pipe which kind of goes out to the person who is breathing, and in the middle of that, there's some contraction that absorbs the CO2. Okay, so we've got a person. Okay, so when the person breathes out, the lid goes up and the trace goes up. And when the person breathes in, the lid goes down and the trace goes down. That's it. Yeah? Okay? Now, there's more, a little bit more to it than that. So this tank is filled with pure oxygen. Okay? And when the person breathes out, the carbon dioxide kind of gets absorbed in this. So even when the person breathes out, it's still only oxygen that's returning into into this tank. Okay? But at the same time, what this does is, it allows us to measure how much oxygen was used up in respiration, because that we'll be missing that amount of oxygen on its way back out again. Yeah? Okay, right, which brings us then to the trace, because it's the trace that's going to be your understanding of what's going on. So, am I even on the screen still? Oh, yeah. Okay. All right, so the trace. Now the trace is usually going to have volume on the y-axis, okay, corresponding to the volume of air. And on the x-axis is going to have time. Okay? Now there's a few key things you need to know. So obviously there's the regular kind of breathing. So the tidal, that's the tidal volume. Okay? If anyone's looking at a trace right now, if there's any measurement that I miss, just let me know. Okay, so you've got the tidal volume, that's your regular breathing. Up, because they're breathing out. Down, because they're breathing in. We good? Okay. Then, you've got this, which is like a maximum forced exhalation. Okay, and then you've got a kind of forced inhalation. So you're kind of measuring the maximum capacity of a person's lungs because of that. Okay? So, yeah. Now, that's the tidal volume, right? From here to here, that's tidal volume. So let's call that 1. 1's tidal volume. What are other measurements? Vital capacity, is that from there to there?
Yeah.
Right, so that, we'll call that, should I label it as something else? Okay, so that is 2, and 2 is vital capacity. All right, what else? Residual volume.
It's from the bottom of vital capacity to the bottom. Yeah.
What's the bottom?
I think just the bottom of the log, right? Yeah.
All the way down to the bottom?
Like halfway between 0.
Oh, right, oh, yeah, that makes sense. Yeah, because, yeah. Okay, the difference between 0, if you squash the lungs completely flat, to how much they have still when you've breathed out completely.
It's from the bottom.
Mm.
Yeah, to the very bottom.
Mm. Okay. Residual volume?
What is it what?
But what? This is when the person breathes out completely, isn't it?
Yeah, that's the inspiratory reserve.
Because, yeah, sometimes, if this is volume in the lungs, then the way we're reading it is completely flipped.
It wouldn't go all the way to 0, like the way I think it's just a bit off.
Yeah.
It's the remaining air inside the lungs.
Yeah, the remaining air in the lungs, therefore then this has to be lung volume. So just be aware of that. Viewers at home, be aware of that. Sometimes you're looking at the volume in the tank, and sometimes the graph might refer to lung volume. If it's lung volume, then everything gets kind of flipped on its head. So when this is going up, that's when the person is breathing out. No, in, because the lung volume is going down. So as they breathe out, the air in the lungs goes down. So yeah, they're breathing in.
Okay.
Yeah, and this is going up because we're looking at lung volume. But if we're looking at tank volume, then that's them breathing out. So just be aware of that. Okay. Any other measures that I'm forgetting?
Let's go to reserve volume.
That's the difference between regular breathing and maximum exhalation.
Yeah, so the top one is higher.
Yeah. Okay. Yeah. So the difference between, oh, this is going to be messed up, but okay. That's the price of reality.
So what did you think I said?
Reserve volume? Inspiratory reserve volume. So
That's just what I said, because that's what I'm usually saying.
yeah, inspiratory. So it's like the difference between regular breathing and how much you could breathe if you forced yourself to breathe in as much as you could. And the opposite of that would be the expiratory reserve. So yeah, so it's whichever way around the graph is, it's the difference between this tidal volume and the maximum in that direction or the maximum in that direction, depending on how you're looking at the graph. Let's call those 4 and 5. Yeah? Okay? All right, last thing on this. It's taken way longer than I thought it would. Last thing. Now, if we are looking at the tank volume, then over time, the trace tends to go down. Okay, if we're looking at the tank volume just for a second. Now, this volume is, the air in the tank is going down because with every breath, a certain amount of oxygen is being used up and expelled as CO2 because of the activity of respiration, right? So because that respiration is happening, now as that oxygen, I wouldn't say gets converted into CO2, but as some of the oxygen gets replaced with CO2, the air in the tank goes down. And so if you work out the slope of this line, gradient, it's actually telling you the rate of oxygen consumption, which kind of parallels with a rate of respiration. Say the person breathes in 10 molecules of oxygen, but they use up 5 for respiration. So when they breathe out, there's 5 molecules of oxygen that return back to the tank and 5 molecules of CO2 that get absorbed in here. If they do that with every breath, breathe in 10, breathe out 5 oxygens, the volume is going to go down. The volume of oxygen in here is going to go down over time. So over time, there tends to be this kind of downward trend. So if you're asked to work out what the rate of oxygen consumption is, you simply have to work out the slope of this line. Yeah, triangles. Yeah, and that's it. Okay, so that's one measurement that you could be asked for. The other one's breathing rate, but I'm sure that we are okay with breathing rate. Yeah? Count the number of peaks in a certain amount of time, and then just extrapolate to 60 seconds or a minute. Yeah?
Yeah.
And seen. Okay. Good job, everybody. Well done, well done, well done.